Hello, I'm Michele Black, Director of Flow Cytometry at Fred Hutch Cancer Center. In this video, I will be covering a cell sorter cleaning and sterility monitoring program developed in my shared resource lab. This video is based on our SRL experience with chronic contamination. Our procedures deviate from the manufacturer user manuals. We have consulted with our field service engineers and tested to ensure the cleaning does not impact normal operation or the integrity of components. Please check with your instrument manufacturer before implementing this cleaning SOP to ensure that it will not void any warranty or service agreement. Cell sorter sterility is critical for protecting sorted samples used in downstream applications. Cells destined for cell culture or in vivo experiments may eventually reveal contamination through visible outgrowth. But even then, antibiotics can suppress that signal, silently skewing functional results without the researcher's knowledge. Cells used in genomic, proteomic, or other molecular assays have no such warning system at all. The presence of contaminating organisms can alter readouts and compromise results without any indication that something has gone wrong. When contamination does occur, the consequences extend well beyond the sample loss. Each incident triggers prolonged decontamination protocol that takes the instrument offline, consumes significant staff time, and creates a backlog that disrupts researcher timelines and facility revenue. Equally damaging is the erosion of trust. Researchers who lose weeks or months of work to a contaminated sort reasonably question whether they can rely on the facility for their most critical experiments. Even a single event can take considerable time to recover from. When I took over as director in 2022, our SRL was experiencing chronic contamination across 7 cell sorters at 2 locations. Staff responded diligently to every incident, following manufacturer cleaning protocols. Additionally, monthly long cleans were performed, replacing the sheath tanks with freshly cleaned and autoclaved ones. Despite these efforts, contamination persisted and would sometimes return within just a few days of cleaning. Because all sorters shared the same commercial-grade sterile PBS, yet contamination never affected all instruments simultaneously, sheath fluid was ruled out as the source. Through systematic trial and error, we identified the true culprits: sheath and auxiliary tanks, fittings, fluid lines, and handling practices during both routine and preventative maintenance. Most notably, dead zones in wet cart fluid lines and auxiliary tanks provided conditions where contamination could persist and biofilms could establish. Standard disinfectants, including 10% bleach and 70% ethanol, cannot penetrate the protective extracellular matrix that defines a biofilm, allowing bacteria to survive routine cleaning and reemerge rapidly. Porous filters and small bore fittings found in instrument wet carts are particularly vulnerable. Understanding this mechanism became the turning point in developing a more effective sterility monitoring and decontamination strategy. To eliminate biofilm contamination from our cell sorters, we developed a 3-step chemical decontamination protocol using the stainless steel and auxiliary tanks. The protocol runs through 3 solutions in sequence: 10% bleach, followed by 70% ethanol, and finally 3% hydrogen peroxide. Between each step, the waste tank is emptied and all fluid lines are flushed with sterile water to prevent cleaning solutions from mixing. The core 3 steps remain the same across all instruments, but the approach varies depending on the sorter. On Fax Aria and Symphony S6s, the auxiliary tanks are not used. For these instruments, decontamination runs entirely through the stainless steel tanks using our aseptic protocol. On the MA900 and the BD Fax Discover S8, the auxiliary tanks and wet carts are required to complete startup and cleaning functions and cannot be bypassed. For these instruments, the protocol extended to include full decontamination of the auxiliary tanks and all wet cart fluid pathways. To address the chronic contamination, at the final hydrogen peroxide step, the solution was increased to 10% and left overnight in all fluid lines throughout both the wet cart and the cytometer. This extended contact time is critical for complete biofilm elimination. The following morning, all lines are flushed with sterile water. Clean, sterilized auxiliary tanks are installed along with an autoclaved sheath tank and new sheath filter. The instrument is then ready to return to service. Once we were able to eliminate the persistent contamination, our routine monthly cleaning follows the same three-step process, with each cleaning fluid contact time only for the duration of the cleaning cycle. Our monitoring practices were also adjusted to better detect even the slightest contamination. We were collecting sterility samples daily and monitoring for 3 to 5 days. Our new protocol extended the period of monitoring for a full 7 days, with observations taken each morning and before the end of each day to allow us to act more quickly if contamination was seen. This allowed us to start the decontamination cycle that evening, letting the instrument sit in 3% hydrogen peroxide overnight. This more effectively addresses contamination while also shortening the downtime the following day. On our Discover S8, we experienced contamination before a single biological sample was run. This contamination was not responding to our cleaning, as contamination events were persistent and becoming more frequent. To track down where it was coming from, we tested all lines in the wet cart. What we observed was that the sheath filter vent line would backflush into the sheath filter when the tank was not pressurized. This line is routed directly to the waste and is not included in any built-in cleaning cycle. Working with our BD field service engineer, we developed a new cleaning protocol that included flushing that vent line. This has now been implemented as part of the standard BD cleaning process for the S8. An important thing to note is that on any sorter, when the instrument is not pressurized, fluid in the lines and fittings can not only harbor contamination, the contamination can grow and migrate over time to other parts of the instrument. When assembling and filling sheath tanks, we try to do so as sterile as possible. All tanks are completely disassembled as part of the monthly long clean. All fittings and the sensor probe are removed, everything gets a thorough wash, and then the tank body and fittings go into the autoclave. The sensor probe is not autoclave compatible, so it goes into 70% ethanol. The tank is placed in the biosafety cabinet for reassembly to keep the components clean and minimize any contamination risk during the assembly process. With the tank fully reassembled, we move it onto a scale. We know the weight of the tank at full volume, so this allows us to fill it accurately without ever having to open it to the room air. For filling, we use a sterile tubing set that we prepare to accompany each 100-liter barrel of PBS. The tank is vented to allow air displacement as fluid enters. We then use a peristaltic pump to drive the PBS into the tank in a controlled, closed system fill. The tank is then ready to be put back on the instrument for daily cleaning and QC. Our sterility monitoring program has been one of the most impactful operational improvements we've made to our core facility. It is built around 4 continuous steps that work together to keep our instruments clean and our users' samples protected. Step 1, monthly and post-service cleaning. Ensure the fluidic system is free from contaminants before samples are run. Step 2, sterility sample collection daily, serves as our baseline for monitoring. Step 3, collected samples are observed over a full 7 days, giving us a meaningful window to detect any microbial growth before it becomes a problem. And Step 4, a robust contamination response to quickly eliminate the contamination, including biofilm clearance, and return the instrument to service. Since implementing our enhanced cleaning and monitoring processes, sorter contamination has decreased significantly to, on average, 1 contamination event per instrument per year. This concludes the video. For more educational content, please visit the ISAC website or social media platforms. Thank you for watching.